Welcome back to the afternoon session here at the day 2 of ARDD. And we have a very exciting session for you now. Our first speaker is Jennifer Elisseeff from Johns Hopkins University. Jennifer, please join us on stage and give her a warm welcome, please. And whenever you're ready.
Great.
Thank you.
Thank you very much. I'm really excited to be here today. I'm not really a standard member of the aging field, so I'd like to give you a little background about my path to get here. So I come from the area of tissue engineering and regenerative medicine, working in biomaterials. We developed these scaffold biomaterials on which we would put stem cells and various biological signals to help regrow tissue. But in the process of our clinical translation, which had been slow in the field, we stopped looking at things like stem cells. We noticed in our first clinical trial that, well, the biomaterials were really redirecting the wound healing process. We looked at these implantable fillers for soft tissue reconstruction, and we saw a tissue-specific immune response that was rather unexpected. So this led us from switching from stem cells to really looking at the immune system as an orchestrator of tissue repair, and that stem cells and vascularization and the matrix growth was really downstream from that immune signaling. And as you may be aware, there's really interesting research on immune system aging. So I'd like to just start with a short summary. As actually a newcomer to the immune field, it can be a little bit daunting with complexity and naming. So we have the innate immune system, which is reacting quickly, usually embedded in the tissue, but it's usually less specific as compared to the adaptive immune system and things like lymphocytes, where you have a slower response, but there's memory and specificity with that response. So some cell types that you may be familiar with, neutrophils and macrophages are of course important with tissue repair and aging, but also looking at T cells, we've been quite interested in that. So we have CD8 T cells, which is important for things like cancer immunotherapy. These cells increase with aging. And these CD4 positive helper cells can differentiate down multiple phenotypes, and we're particularly interested in the Th2 and Th17 pathways, which I'm just going to mention a little bit more. So for infection and immunotherapy, the Th1 differentiation pathway is important, and interferon gammas, I think there's another talk on that. Th2 has generally been associated with helminths, infections, allergies, and also healing. And finally, Th17, again, connected with infectious responses, but also fibrosis. And I'd like to point out these sort of negative regulatory aspects. So if you have Th17, you're blocking this, and they sort of regulate each other. And so that'll be important as we talk about their role in aging. So the immune system in tissue repair, sometimes it helps and sometimes it hurts, right? So things like inflammatory cells can impede, CD8 T cells can impede tissue repair, and type 2 signals connected with IL-4, IL-5, IL-13 are connected with tissue repair in muscle, in the liver, and even the central nervous system. So from the biomaterial side, looking there again, there's some matrix scaffolds, which you could argue are sort of a mimic of damaged tissue to promote the tissue repair response. They're used clinically in a number of reconstruction applications. So we looked at these in a muscle wound, and we found that these scaffolds upregulate T cells, helper T cells, making interleukin-4. This has an impact on, of course, other cell types, such as the macrophage. And also importantly, there are systemic changes. So if we look at the draining lymph node and even the distal lymph node, we see changes in these cytokine expressions. So these biomaterials can enhance these responses. So I know we're in an aging group and people like things like mTOR, so I wanted to just mention how that connects. So if we take a RAG knockout mouse that doesn't have T cells or B cells, you limit the repair. You see this fibrosis. If we adoptively transfer healthy wild-type T cells, we can get nice repair. But Jonathan Powell was next door to me for a little while, and he had these Richter knockout mice. So we're manipulating this TORC2 complex. If we transfer instead Richter knockout T cells, what we get is fibrosis and adipogenesis, or dysregulated tissue repair. Now, what happens when we put these materials in older mice? Well, you can see it looks quite different. We have a lot more adipogenesis happening and a lot more fibrosis. And if we look at the actual muscle tissue, we have nuclei. This is dystrophin, and the nuclei here are still on the edges of the muscle fibers. And that's symbolic of not very inactive cells. So when you have repair, you tend to see the nuclei in the center. So something's wrong here. Increased fibrosis and increased adipogenesis. Now, coming from the engineering side, we like models. So we actually have some nice models for fibrosis. Any implant made of a synthetic material, when placed in the body, will induce fibrosis. And one that we can get a lot of samples from are breast implants and exchanges for reconstructive surgery. So they have this fibrous capsule around the implant, and they're being exchanged frequently during the reconstruction process. So we can take that capsule around that implant and look at what cells are there. Now, macrophages and neutrophils were known to be a part of the foreign body response in fibrosis, but T cells less so. However, we see a large number, and they're predominantly making IL-17. And that IL-17 is coming from both conventional helper T cells and gamma delta T cells. Now, we are making a connection with senescence here, which I'll talk more in a little bit, but essentially this is showing around those implants that we have IL-17 being expressed, and then P16, a senescence marker, and there's a correlation with the levels of RNA. So looking at preclinical models, we have some materials that we can implant in a mouse, again, to induce this fibrotic response. And we see a similar trend that when you have the implant here in red, you see increased levels of IL-17 compared to no implant. We also see P16 increasing, so you have an early damage response increase in P16 that happens, you can see at 1 week here. And then at 6 weeks, we see a big difference with that implant that's inducing fibrosis. If we sort out the stromal cells and get rid of the immune cells, we can see an even more significant increase in that P16. So if we look at a wild-type animal, you see P16 staining in that fibrotic area. If we knock out IL-17 that's being made by these T cells, gamma delta T cells and conventional T cells, we can reduce that senescence. So that started us looking in this connection between immune response and immune phenotypes and senescence. So if we take a senescence fibroblast that we've artificially induced senescence in vitro and culture them next to naive T cells, we see a significant increase in IL-17 being produced by those T cells when TGF-beta is present. If TGF-beta is not present, it's actually interferon gamma. We can look the other way around too. We can artificially skew those T cells towards a Th17 pathway and culture them next to healthy fibroblasts and induce signatures of senescence, P16, P21, and IL-6. And we characterize this, the phenotype more broadly, as an inflammatory induced senescence. But what really is a senescent cell? This suggests another way that they can be formed, but what are they in vivo? We've just been looking at in vitro. Well, again, we can use this model system to try to understand that. So using a P16 cream mouse developed by Darren Baker and Jan van Derzen, we were able to track these cells with TD tomato expression, looking at when that fibrosis develops. So after the injury is healed from the implantation and when the fibrosis is starting. So we could see small number of cells, but still there when we have the induction of that reporter. Which cells are senescent? So as suggested by the earlier data I showed you, it's non-immune cells, although we do have a few showing here, but majority are those CD29 positive, CD31 negative, CD45 negative too. We can take those cells then and sort them out. So we're taking those CD29 positive cells, sorting them out, and we can do some bulk sequencing. So we really get in-depth expression profiling information. What we find is when we look at some gene set enrichment analysis, we see a number of vascular-related genes and actually, strangely, chondrocyte differentiation-related genes. And I used to work in the cartilage field, so we thought, well, maybe this is due to contamination with some tendon or something. But actually, it's not. So there's some interesting similarities between that fibrosis matrix production and what you see in cartilage tissue. Now, when you do single-cell analysis, single-cell sequencing of those same CD29 positive cells, you can get a number of different clusters. But again, as you may be aware of, you don't see things like P16, and P21 is not very specific. And you also saw that it's a really small number of cells, so you're really not going to get those senescent cells separating out computationally as a cluster. So what we did is we developed a method to take that bulk sequencing signature and apply it to the single-cell data so then we could pick out which ones were senescent. So we developed this senescent signature. So looking at each cell and then each gene, is it similar or different than that bulk signature? And so you can see we get this heat map of which cells appear to be senescent in that population, and you can see which clusters are enriched. So as suggested by that gene set analysis, there's a pericyte population and the more fibrotic type population. You can do a mouse to human transfer, so this is the same population with the same fibrotic tissue from an implant. And you can see which clusters have that strong senescent signature. And you can see what genes are driving those similarities. So the pericyte-like cluster, you can see interesting things like Notch3 playing a role here, and just throwing out CCN here, that's a mechanoresponsive gene, so the interesting thinking of fibrosis and the mechanosensitivity, and a lot of matrix production in that fibrotic cluster. So you can see that also morphologically, so the P16 staining in purple here, you see associated with blood vessels, as you would expect a pericyte. And then you can see also some P16 cells with some very typical fibroblast morphology. So there are plenty of programs where you can pull out secreted factors from those clusters. So here's some examples of some SASP factors or secreted factors from those different clusters. And you can see which ones are specific and ones that might be overlapping. Something to keep in mind is that this cancer data set, for example, very, very large data set. So you have a lot more cells in that compared to, let's say, our muscle injury model. But you can look at any of the data sets with this senescent signature. Now, speaking of SAFs, you want to know who these cells are talking to. So we developed a program that actually looks at transcription factor activation and receptor ligand analysis. And we can look at the connections between these different senescent cells and, in particular, immune cells. I won't get into the details of this because of time, but what we find is that that pericyte senescent cell is connected with, communicating with macrophages, and then you have another set of macrophage populations that are communicating with those fibrotic senescent cells. So you can look at that data set, it's available online, and look for your sort of cells and genes of interest in those communication pathways. So for the end here, I'd like to sort of combine it all to look at that aging environment that I showed you at the beginning that was not repairing when we put that pro-regenerative material in there, but was instead undergoing fibrosis and adipogenesis. So I said IL-4 was important in that regenerative response. What happens with aging? Well, in the old animal, you get a decrease in that IL-4. Well, if that wasn't bad enough, you also get an increase in IL-17 in that muscle tissue. So why is that happening? So if we look just at some immune organs and how they change with aging, we can see a few things. So it is well known that naive cell numbers, naive T cells decrease with aging. But it's sort of interesting there's some tissue specificity there. So the spleen, we see a significant decrease. Still significant in the lymph node. This is the inguinal lymph node, but not as much as the spleen. So as the naive cells go down, you can imagine the effector cells are going up. So both in the lymph node and in the spleen, your T cells have an effector phenotype. What specifically? Well, if we look at gamma delta T cells, the old animals significantly decrease interferon gamma expression, but have a lot more IL-17. CD4 T cells, again, you've got actually both increases in the inflammatory interferon gamma and the IL-17. So that's before any injury happens. What's interesting, though, is injury also uncovers age-related change. So we did some protein mapping to sort of look at other genes, to find which other genes might be interesting to look at. What I'd like to point out in these is saline is injury alone, and then we have our implant. If you look at no injury, in most of these, there's not much of a difference between the young and the old. But the injury response really changes with the young and the old. And you see these inflammatory genes going up with that injury. Now, for those who like macrophages, I don't want you to feel neglected. So single-cell comparison of the young and the old. The older tissues have much fewer activated myeloid cells, a lot less antigen presentation happening, in addition to that fibrosis. But aged T cells are different. So if we take young and old spleens and we pull out the naive T cells, yes, there are fewer naive T cells in the spleen, but we can isolate them so you have the same amount. We put them in culture conditions to push them towards this Th17 phenotype that we think is important here. Are creeping in, but with differentiation conditions, those T cells to differentiate. And which way are they differentiating? We see significantly more increase in the ROR-C differentiation.
We will change it for number 12.
My batteries are out, I guess. Okay, so you get more, these cells that, they all started off as naive. In culture, they can differentiate more towards that pathway. But here's the stuff that I found most interesting. If we take the media of what those T cells are making, so they're both going down the Th17 pathway, but the old ones are making different things. So in particular, I was quite intrigued by the vascular factors. So you have increased VEGF. So the red here is the old animals. Increased VEGF, LIF, and a number of these factors. So IL-17 is sort of similar. So if you use your standard assays, you might not see a difference between the old and young. But all these other factors are different. Aged senescent cells are different too. So if we use that transfer learning technique that I showed you before, this population, this cluster, we found to be, have a higher senescent signature. We still see the endothelial parasite signature of senescent cells, but this fibroblast one is different. And we can look at the SASP from those parasites and fibroblasts between the young and the old, and there is definitely some overlap between the two. But then there are also unique factors expressed by the young versus old senescent cells. What happens when you put this together and look at how they're communicating? Well, the young global signaling network, you can see a lot of communication between the tissue module, fibroblast module, and antigen presentation. But when we apply dominant to the old animals, much more limited communication between the young and the old. So what do we, when we look specifically at ligands and receptors, if we pull out those endothelial cells and pericytes and the senescent fibroblasts, there's a little bit of communication in the young, right? Probably important for regeneration, these muscle cells and the endopericyte, but not much going on here. But look in the old animals. T cells are very active, and these senescent fibroblasts, a lot more activity there. And you can pull out the genes, but I don't have time to get into that. So what we're looking now as a model is that you have these senescent vascular-related cells connected with angiogenesis, sensing things like hypoxia, and then you have these more matrix-associated or more fibroblast-like senescent cells that are making matrix, and the immune system plays an important role of integrating that communication. So aging and tissue repair. Tissue damage has many sources. Infection causes tissue damage, in addition to basic trauma, and I would argue basic organ functioning can cause tissue damage. So when we think of therapies for repair, we need to consider the aging background for efficacy, and then the immune aging phenotype, for example, the Th17 phenotype, it actually negatively regulates and blocks Th2 and the type 2 immune system that's important for regeneration. So it's not necessarily that your stem cells are old, just you've got factors that are actively inhibiting the repair process with aging. So I think my time is up, and I'd be happy to answer any questions. Thank you.
Thank you so much for a great talk, Jennifer. Do we have any questions in the audience? I could ask the first one. So you see this exacerbated response to injury. Is that also temporal? So is it like, does it last longer, or is it more difficult to shut down, or?
Well, usually with fibrosis and even adipogenesis, you do see some chronic inflammation even. And when you look at the, for example, P16 expression in the young animals, you have some residual P16 in senescence even at 6 weeks. So it's definitely longer, but I would say it's not even fully resolving because of that chronic inflammation. that's generally associated with fibrosis.
Right. Okay, very interesting. We have a question here.
A very naive question. So PD-L1 and CTLA-4 inhibitors are very commonly used in, clinical oncology. So in terms of aging and, T cells, what you found, so do you think they would be beneficial or detrimental in terms of, battling aging?
I'm sorry, can you repeat the first part?
PD-L1 and CTLA-4 inhibitors. So can you, comment on T cell and aging? Yes.
Yeah,
Thank
so
you.
we're actually just in the process of looking at checkpoints in the fibrosis and wound healing process, and it does look like those checkpoints go up, right? So as a part of the normal regulation of the wound healing process, I think the checkpoints are actually important in that resolution and dampening the immune response as healing occurs. But then we're quite interested in when it is non-healing and chronic, we do see that high checkpoint expression. My concern is, though, that you could potentially keep that sort of bad inflammation happening or even increasing if you give the checkpoints. But we're doing the same type of analysis actually in the tumor microenvironment and looking at that fibrosis that's actually associated with immunotherapy non-response. And being able to do combination therapies, actually, we've been targeting IL-17. So you give the pro-regenerative material and block IL-17, and you can get a response in the old animals. That's the slide I skipped over for a time.
Very cool. Thank you so much, Jennifer. Amazing talk. And remember, if you have more questions, you can post them on Slack.